Hi, Michael. Thank you so much for joining me today.
Sanjeev, my pleasure. You look very official with the tie and jacket today.
Thank you so much. I thought it'd be best our viewers should definitely hear from you because you're an expert in the whole field of aging for the last, I think, at least 30 years. And you've written a book.
Five, actually, but yes.
You've written a book on this.
Several, actually, yes.
And numerous articles. So I thought, who better to do that? And I'm learning so much from you. And I thought our listeners should also to get a chance to hear from you. So maybe before we start, do you mind like taking us a little bit through your background? Like, how did you get involved in the space? Why is it so important to you? That would be great.
Well, I first got intrigued by this back 45 years ago, actually, at Stanford. I was finishing a PhD in neurobiology and was teaching in the medical school. Mm-hmm. In fact, they paid for me to get the MD, which was very convenient. But I was originally working on the development of the nervous system, which I find awesome. Absolutely, this floors me. But I found, and most people felt the same way. But I found that whenever I talked about aging and the dissolution of the nervous system, for example, with the dementias, people would sort of shrug and say, well, of course, it just happens. It's wear and tear, it's entropy. You get old, you rust, things fall apart, what do you expect? And I found that a very blasé attitude. And I find that usually when people get very blasé about things and think they're not complicated, it means they haven't tried. Almost everything in the world I look at is more complicated than I think it is. And that's very much true of aging too. And the more I looked into it, the more I discovered that there was a lot more to it than it just happens. It's just entropy, it's just wear and tear. Mm-hmm. It was complicated. But the problem I had initially was that there were all these little things, sort of like the blind man and the elephant. There were what we knew about mitochondrial dysfunction, Hutchinson-Gilford progeria, right, and genetic changes, and the interesting phenomena of germ cell, lack of aging in germ cell lineages, all those things, fascinating. But I couldn't think of where to get involved from a research perspective. So I ended up in clinical medicine, which I enjoyed thoroughly in an emotional context, but my intellectual love remained with aging. And then in the early 90s, I ran into some of the earliest data. In fact, I used to have it all under my bed on cell aging, telomeres. And I began to see from some of the early conferences how you could begin to put all the different pieces of the elephant together into a unified model. And that's what got me intrigued by it. In those days, actually, Geron Corporation flew me out to California and gave me access to some of their proprietary data because they were doing some fascinating work. And then 25 years ago this year, I gave the first talk ever at the National Institutes of Health on the prospects for reversing the aging process. And what I pointed out was that if we can truly overcome our assumptions and look carefully at what's going on and actually intervene in the fundamental aging process, then the potential is to be able to cure and prevent age-related diseases. But it's more than just saying, let's treat the Band-Aids, the biomarkers, the downstream outcomes, the signs, the symptoms. I think we really need to understand it. And so I got involved in that, wrote the first articles ever in the medical literature on this in JAMA back about 23 years ago and 21 years ago. Mm-hmm. And then wrote the first textbook on this with Oxford University Press. And then slowly have gotten, as well as writing numerous other articles and a more recent book that was the Wall Street Journal thought well of, and so did the London Times, meant for the general public as opposed to the academic book. I've gotten involved in actually taking this to human clinical trials, and that's where we are now. If we look back at the data, what you'll find is that we first demonstrated we could reverse aging in human cells now 21 years ago, 23 years ago, and in human tissues 21 years ago. Most people are entirely unaware of that. And then to a degree in actual whole organisms, now about 10 years ago. And we're not very good at it, but we're getting better. So it's a remarkable time, but I think one, most people have no idea that these things are being done, that they're feasible mm-hmm. and in progress. And two, most people actually do not understand anything about aging. They think they do. It's like, it's like 406 years ago, the Vatican took Galileo to task because they knew that the Earth was the center of the universe and Galileo knew it wasn't. And so he ran into huge trouble. But the problem wasn't just that people didn't have access to the data. The problem was the interpretation of the data. And the Vatican started with the assumption that the Earth was the center of the universe and they weren't going to change it, which is another example of why science is not a democracy. It just
right.
is. And as I say, Galileo and Copernicus before him and Kepler before knew otherwise. But that's kind of where we are now with regard to aging. Most people just assume they know what it is. It's entropy. And entropy is a part of it, but it's not just entropy. It's entropy versus maintenance, for example, and then it gets extraordinarily complicated.
By entropy, you mean like wear and tear? Like, what do you mean by entropy? Like
Yes, exactly. It's sort of, in this case, a generic term for wear and tear. So I'll give you a sort of a funny example. Why is it that my skin ages? Well, wear and tear clearly plays a role. If, for example, I expose it to a great deal of UV radiation, it ages faster. If I undergo a great deal of trauma, it ages faster. On the other hand, we also know that your body maintains itself. So why is it that the same degree of trauma doesn't age a 4-year-old as fast as it does a 40-year-old or an 80-year-old? And that begins to show there's some of the complexity involved. Another example might be, a lot of people would sort of wave their hands and say, well, entropy, wear and tear occur to mitochondria. And so we age because the mitochondria fail.
Right.
Except that every mitochondria in your body, and everyone who's listening to this, derives from your mother, who got it from her mother, so it was already several decades old, who got it from your grandmother, several more decades. And that line of mitochondria, depending on who's making this estimate, because there are several of them, go back just short of 2 billion years. 1.7 is one of the current estimates from the age at which we began to have eukaryotic cells import the organelles of mitochondria. But the point is, every mitochondria in your body showed no evidence whatsoever of aging up for several billion years, to the point you inherited it from your mother, and yet in a matter of decades, you undergo entropic changes, or apparently entropic changes, to your mitochondria. So if you want to say aging is just mitochondria, waving your hands, you're going to have to explain why it doesn't occur for a couple of billion years and then does. It's not that entropy doesn't play a role, but it's clearly more complicated than it's just entropy. It's just not.
Mm-hmm. So this, what you're saying is that we inherit our mitochondria from at least the DNA from the germ cells. No, is that
the actual organelles? Now,
the actual organelles?
If you're looking at your DNA, if you're looking at, for example, the germ cells themselves,
yes,
those go back, on the estimate several more billion years before that. So you were something in the order of 3.5+ billion years old in the sense of a cell, going back prokaryotic and beyond. And that cellular line, in retrospect, showed no age-related changes. When you inherited it, you inherited a cell, in this case, a fertilized ovum, that had no evidence of age-related changes, and yet it does over your few decades. And the same is really true of your mitochondria. You inherited essentially flawless mitochondria. So if you want to say that mitochondria cause aging, you'd have to explain why most mitochondria retrospectively didn't age, and now they do. I'm not saying entropy doesn't play a role, but it's more complicated than it's just entropy and a lot of hand waving. It's complicated.
So it sounds like there's some type of switch perhaps that goes off that perhaps turns the aging process on, and because you see in other animals, some seem to live very long lives for whatever reason. And so do we understand what is this switch?
I think we do if we look at it more carefully. And it's intriguing what you say about that. For example, I'm just thinking about lifetimes. Human beings live about, on the average, 3/4 of a century. Gorillas, a little short of half a century. Most domestic animals, a little short of about 2 decades, or a little more in some breeds. And most laboratory rodents 1 to 2.5 years, depending again on the particular breed involved. And you might say, well, why do they age at different rates? Well, clearly it's not just entropy, because something genetically is going on. And again, hand waving. For example, if I take a typical laboratory mouse, I share about 75% of the protein expressing genes. I'm one of the regulatory genes. And most people, again, would wave their hands and say, well, obviously it has something to do with that 25% I don't share, which is true, but it doesn't get you anywhere. It doesn't tell you what the process is that does that. And even when you look within a particular species like human beings, we know that Hutchinson-Gilford progeroids live an average of about 12.7 years, die overwhelmingly of what appear to be age-related diseases, typically age-related vascular disease, stroke and heart attacks kill most of them. And I used to gather them together from all over the world about 2 decades ago. And again, you could say it's genetic. And in fact, we know the laminate gene plays a role. All right, but what is that role? Why precisely do you have a laminate gene that causes aging in progeroids, and I, who don't have that same allele, live 4 or 5 times as long? Why does that happen? So ascribing it to genes It's not an explanation. It simply tells you where you should start looking. In this case, with Hutchinson-Gilford's, the question is, what does that allele do that's different than my allele? Why is that happening? And the same thing is true of aging in general. The same thing is true of the question that we've been discussing, which is, why do we find that some mitochondria retrospectively didn't age? Other mitochondria do. Some age faster than others. Why is that? Again, if you're going to talk about what aging is, you cannot simply wave your hands naively and say, it just happens. It's entropy, it's wear and tear. It's partly that.